Hello, everyone. Welcome to our session on allelic interactions and non-allelic interactions in gene interactions. So, yesterday we missed a particular point in codominance. So, we'll first discuss that point and then we'll move on to the lethal genes. So, the point was nothing but an example of codominance, which is nothing but uh, the ABO blood groups in man. So ABO blood groups in man, which will have a detailed study in multiple alleles. So this is also the blood grouping in man is also taken as an example of uh, codominance. Example in the sense, so codominance, as we said yesterday, here both the alleles will be expressed at the same time. That means there will not be expression of one allele and the repression of the other one. Such a type of relation or a mixed character will not be expressed in such cases. In codominance, both the alleles can be seen in the pure form as the phenotype. It will not be a mixture color like the pink flowers or etc. You can separate both the alleles. You can see both the alleles in separate forms or distinctively in the heterozygote. That is the main difference between our incomplete dominance and codominance, where the individual or independent expression of both the alleles can be seen in the heterozygote. But when you come to incomplete dominance, you can see that there is a mixture because both the alleles they get mixed up and a mixed expression or an intermediate expression can be seen in the case of incomplete dominance. But here, each allele is independent of its own and it expresses its own feature. So that both the phenotype or the phenotypes of both the alleles can be seen in the heterozygote. And one such an example or a wonderful example is the ABO blood group in man. So ABO blood group in man is controlled by mainly by three alleles. That's why we'll detail that in multiple alleles. We'll see a detailed study or we'll come to a detailed discussion of this particular ABO blood group system in multiple allelism. But for the sake of example, in codominance, we'll be discussing how codominance or what is codominance in blood grouping. So, blood group in man is determined by three alleles uh, rather than our examples with two alleles. This one is controlled by blood group in man is controlled by three alleles. As you can see here, one allele is IA, the other allele is IB, and the other allele is IO. Here it is written as small I. It is also written as IO. So there are three alleles for a particular gene. That means for blood grouping or to determine blood group, to determine a particular trait, rather than one or rather than two alleles for a gene, here you are having three alleles for a gene: IA, IB, and IO. And it is in this particular case, this IA allele, the first one, and the second one, the IB allele, these both are dominant. They are dominant over the recessive one, which is IO or small I in this particular diagram. So what happens is this IA and IB being dominant, when IA and IB comes together in an individual, they express together. Their expression can be seen together, and that is what is called as codominance. So that is what codominance, that is the IA as well as IB in the case of blood grouping, that is in AB blood group. Both the allele, that is allele A and allele B, are codominant, or they express both the antigens on the surface of the RBC, and they are said to be codominant. So the example of codominance in blood grouping is the AB blood group of man. In that allele A and allele B, it expresses both the alleles are expressed in that particular case. But you can see here in this table, when the IA allele is with small I or IO, the blood group is A because this one is recessive. And when IB is with the recessive one IO, that blood group is B. That's why we say that this IO or small I is recessive to both IA and IB, and when they come together in an individual, they have a co-expression or co-dominance. Both expresses its phenotype, and that is what is called as co-dominance in blood group. We'll study the details of what is blood group, etc., in multiple allelism in our discussion in multiple allelism. So now we were discussing about the variations in dominance, and we are moving on to the last variation in dominance, and that one is lethal genes. So coming to lethal genes, an allele can is capable of causing death of an organism is called as a lethal gene. So from the name itself, it's evident. Lethal means it is something which causes death. So an allele or a gene which is capable of causing death is usually referred to as a lethal allele or a lethal gene. And there are certain genes which control phenotypic trait. They have two functions. That means they have the ability to produce a phenotype, and at certain times they cause or they determine the viability of that particular individual. Such genes come under the category which are called as lethal genes, and these lethal genes are usually are useful in determining the developmental stage at which the gene is normally acting. That means whenever this gene starts to act differently, that results in the death of the individual, or that causes a threat to its viability. So it is such genes are usually used to study the developmental stages where some genes are getting activated, etc. And mainly there are to classify lethal genes, there are mainly four types of lethal genes are there, but as per your syllabus we have only three, but we'll discuss all the four in this particular discussion. And they are, you have the dominant lethal genes, the recessive lethal genes, the sex linked lethal genes, and the fourth one is there which is called as the conditional lethal genes, that will also be discussed under this particular session. So we are now discussing the variations of dominance in allelic interaction. So these are all allelic interactions, and in that we are at the last one, which is called as the lethal gene. So we will move on to the first one, which is called as the dominant lethal genes. So, dominant lethal genes are the genes that cause lethality in the homozygous dominant condition. That means whenever they are present in the homozygous dominant condition, they cause a lethality. And the example of this is the creeper lethal inheritance in chicken. So, here you can see the diagram. So, creeper, or this particular gene, which is called as a creeper lethal gene, uh, as said above, when it is present in the homozygous condition, it causes the death of the particular individual in the embryonic stage itself. And when it is present in the heterozygous condition, that means when the dominant gene is present in the heterozygous condition with the normal allele, what happens is you get such a type of phenotype which is having short limbs. That means, as we said before, in the beginning, in the introduction of lethal genes, we said that these lethal genes, they have the ability to produce a phenotype, but at certain times they cause lethality. So the phenotype that is produced by this particular lethal gene is short statureness. That means the limbs will be short. Limbs in the sense both the hind limbs as well as the forelimbs. In this case of a creeper, the wings as well as the legs will be short. So this is a creeper one, and this one is creeper. This is a normal female and this is a normal male. So this is a creeper male and this one is a creeper female, and both these are heterozygotes because this particular gene expresses only in the heterozygous condition. In homozygous dominant condition, it will cause lethality. So here the creeper allele causes the development of short and standard limbs, and creeper is a dominant gene. When present in the homozygous condition, it is causing death in the embryonic stages. The heterozygous display the phenotype. So you can see this here. So this is an X-ray image of a embryo, where you can see that this will not develop further. It will die in the embryonic stage itself. You can see the limbs are very poorly developed, or you can say that the that only the limb buds can be seen. As a result, it will not develop, complete its embryonic development. It will die in the embryonic stage itself, and this is what is called as a lethal one, which is homozygous dominant. When it is heterozygous, you call that as a creeper, because that is the one which we see in the living form, the creeper form, this one and this one. So the creeper, you can see they're having short limbs, both the forelimbs, the wings as well as the hind limbs, that will be short statured, and this is a normal, the wild type one. So what happens here is, as this one will not be seen, because when you do a cross, this one will not be counted because this is not coming out of the egg, or this is not coming out live. You will not see this particular one, the lethal one. As a result, the normal ratio, that is a normal ratio of 3 is to 1. that is a normal monohybrid ratio of a Mendelian ratio 3 is to 1, that will change to 2 is to 1. Because this one, if you see this table, this one is lethal. You get only two creepers and one normal. That will be seen, or that is the one what you see as a phenotype. The other is dying in the embryonic stage itself. As a result, the normal 3 is to 1 ratio or 1 is to 2 is to 1 ratio, the phenotypic ratio 3 is to 1, and the genotypic ratio 1 is to 2 is to 1, that changes to simply 2 is to 1. Both the phenotypic as well as the genotypic ratio will be 2 is to 1 in the case of the dominant lethal genes, and that will be 2 by 3 creepers and 1 by 3 normal. You cannot see the four, that means 1 by 4, 2 by 4, and 1 by 4 was a normal ratio, but here it is changed to just 2 by 3 and 1 by 3, because only three individuals are born live, the other one, this one is being lethal. The total number is reduced to 3 from 4. So this is what is called as lethal gene, and the example is the creeper lethal in the case of chickens. So coming to the second type, which is the recessive lethal genes. So this causes the death of the individual which carry both the copies of the lethal gene. So as we discussed earlier, for recessive alleles, whenever it becomes deleterious, recessive alleles are expressed only in the homozygous condition. Only in the homozygous condition they express or they cause their particular phenotype. And here, lethality of this recessive lethal gene is caused only in the homozygous condition. In the heterozygous condition, they express a phenotype. So, in the case of this coat color in mice, you have a wild type coat color, which is called as the agouti or black coat color, and you can see the dominant one, which is called as the yellow. And in this case, yellow is said to be dominant because you can see that the yellow fur color or the yellow coat color is seen, or that is that particular gene expresses both in this heterozygote as well as in the lethal ones. In both cases, the coat color, the gene which causes, or the gene which is responsible for these three, is the yellow or the AY gene. So here it is this AY gene that gives you the yellow color. Here, because it is having two AY gene, this yellow coat color is considered to be dominant in this particular case. It is the yellow coat color is found to be, or it is said to be dominant over the wild type, which is called as the black one. And the examples of the human disease caused by the recessive lethal genes are the cystic fibrosis, sickle cell anemia, and also achondroplasia. So these are the examples of recessive lethal genes. Of this, achondroplasia is an autosomal bone disorder which results in causes dwarfism. This achondroplasia comes under recessive lethal gene in the sense that when achondroplasia gene is present in the homozygous condition like this, it causes dwarfism or not. When I say dwarfism here, don't consider every dwarfism as achondroplasia. This is a special or a this is a genetic disorder which is caused by an autosomal dominant gene. Dwarfism may be also due to hormonal disorder, so don't take every dwarfism as achondroplasia. So this is a special genetic disorder, achondroplasia, which one of its feature is dwarfism. While the inheritance of one achondroplasia, that means whenever it is present in this particular form, that means when achondroplasia gene is present in the homozygous condition, that will be lethal because it is a recessive lethal gene. But whenever it is in the heterozygous condition, what happens is it results in dwarfism. And another example is this one, the coat color in mice. Coat color in mice, as I said earlier, the yellow coat color is dominant, and when you cross these two yellow mices, what you get is rather than a 3:1 ratio. When you cross two heterozygotes, rather than getting a 3:1 ratio, you get a 2:1 ratio. It is same as that of the dominant lethal gene, where you get 2 by 3 of this and 1 by 3 of this. And one quarter of the offsprings from crosses between heterozygote died during the embryonic stage due to the lethal genes. Recessive lethal genes can also code for either dominant or recessive trait. So this is another point that is to be noted in recessive lethal genes. So we'll explain this. So recessive lethal genes can code for either a dominant or a recessive trait. So what I explained here, the code color in mice, as we discussed earlier, in this recessive gene codes for a trait which is dominant. As we said earlier, this is a dominant one where this achondroplasia gene, because this particular thing is being expressed in all these three, even in this dead mice also, this gene is the one which is causing lethality. As a result, you call this one as a dominant gene. The same is the case with achondroplasia gene also. It is an autosomal dominant bone disorder, in the sense the achondroplasia gene, because achondroplasia gene is expressed both in the heterozygote as short statedness and as lethal when it is homozygous. That gene is considered to be dominant over the normal wild type allele. That is why we say that recessive lethal genes can either code for a dominant trait or a recessive trait. The examples which we discussed, both the case with mice as well as achondroplasia, are the examples of the dominant, the recessive lethal gene coding for a dominant trait. So this is the example which is to be discussed in detail, which is cystic fibrosis. So cystic fibrosis is an example of recessive lethal gene. This is a genetic disorder which is inherited in an autosomal recessive manner, in the sense the disease will be expressed only in the homozygous condition. In the heterozygous individual will be normal. And this cystic fibrosis is an example of what we said earlier, that is the recessive lethal gene which is coding for a recessive trait. So the recessive trait here is the mutant one which is causing the disease, but here the heterozygote as well as the homozygous dominant is the that causes that will be normal. Homozygous dominant and heterozygote in this case will be normal without this particular disorder, which is called as cystic fibrosis. But the other one, that is homozygous recessive, that will have the condition which is called as uh, cystic fibrosis. So it is an autosomal recessive, it is inherited in an autosomal recessive manner. Autosomal recessive means it is carried or it is a defect of an autosomal gene, not the sex chromosome, of the 22, 1 to 22 chromosome. One of the autosomal chromosome disorder results in cystic fibrosis, and that it is inherited in a recessive manner. In the sense, I stated clearly that only in the homozygous recessive condition, this particular gene or this particular disease will be expressed. Both the heterozygote and the uh, dominant homozygous dominant will be normal individuals without the disorder. So this is caused when both the copies of this particular CFTR gene, which is cystic fibrosis transmembrane conductance regulator, it is a transmembrane channel which is seen in our skin, in our epidermal cells, especially that lining the mucous membrane. Those epidermis or epidermal lining or epidermis that is lining the mucous membrane, for example our respiratory tract, then our gastrointestinal tract, our intestine, then certain glands, etc. All the lining such mucous membranes will have this particular channel, and this is responsible for the production of mucus and maintaining the mucosal layer, etc. This particular receptor or this particular channel, which is called as cystic fibrosis transmembrane conductance regulator. So when there is a defect in this, in normal individuals, the mucous membrane will be normal, it will function normally, etc. Whenever there is a defective, that means heterozygotes as well as the homozygous dominant will be normal without any issues. But when you have a homozygous recessive condition for this particular gene, which is defect in both the alleles, what happens is these conductors or these receptor or these particular transmembrane channels, they do not work, and that will result in the improper functioning of the mucous membrane. The lining will not be containing proper mucosal layer, because there is a particular consistency for the mucosal layer to function properly. It should be in a, very thin and semi-fluid nature, so that all the bacteria or the particles which come in will be trapped in our respiratory tracts, which prevents infection, etc. So what happens in this case is that the mucus becomes thick, As a result, it will not have the proper function. And such individuals will be prone with many, always they will be prone with lung infections, then chest congestions, etc. And they will also be affected by, as I said, it is related to mucous membrane. Wherever you have mucous membrane, such areas will be affected, like the liver, the intestine, the pancreas, the sweat glands, etc. So all this will be affected because of this CFTR gene, and they suffer mainly. They have the difficulty in breathing, and also they will have frequent lung infections. And there is no known cure for cystic fibrosis. They have to live with cystic fibrosis for their lifelong, because and usually it is said that they are severely prone to infections like pneumonia, etc. That might be the cause of their death. So this is also an example of a recessive lethal gene, which is lethal because such individuals, they do not have more life expectancy because of such recurring infections, etc. So that's with our recessive uh, lethal genes. And the next one is the third one, which is sex-linked lethal genes, which is also called as sublethal and semi-lethal genes, that we'll discuss in the next session. Thank you for hearing.